Hello, this is Yashveen. In this video, I'll be solving paper 2 variant 2, February-March 2021 9700 biology. Starting with question number 1a, figure 1.1 is an image of a transverse section through the trachea of the gas exchange system, where you have a few labels. Write the names of the structures X and Y in figure 1.1 in the boxes provided. In the structure of the trachea, going from the lumen inwards, first you'd have the ciliated epithelium, then the columnar epithelium where you'd have long cells, which is not shown in the diagram over here, and then you'd have mucus glands, so that would be X, and then there'd be this layer of cartilage, which is Y, which would be surrounded by smooth muscles, and that are also not shown in this diagram. But here you can write down the labels X as mucus glands and Y as cartilage. B. The perichondrium contains collagen fibers composed of collagen molecules. Collagen is an example of one type of biological molecule. State the name of the type of biological molecule of which collagen is an example. So this collagen molecule is an example of a protein. C. Some structures in the gas exchange system are listed in alphabetical order in table 1.1. Write yes in the box provided if the structure contains smooth muscle and no if it does not contain smooth muscle. An alveolus only has walls of squamous epithelium, so there's no smooth muscle in alveoli, and all of the rest of the structures do contain smooth muscles which are required for their movement, for the movement of these structures during the breathing process. Two starch molecules are the main storage molecules in many types of cereal grain, such as the grain of the barley plant. A. When the seed inside a barley germinates, gene coding for the digestive enzymes are switched on. The enzymes that are synthesized catalyze the hydrolysis of storage molecules, such as proteins and starch. 1. Explain what is meant by a gene. So the definition of gene is a length of DNA that codes for a polypeptide. So the DNA contains triplets which would be used to transcribe mRNA, and that would be used for translation, along with tRNA, which would bring amino acids to join and form a polypeptide. The hydrolysis of proteins in the barley seed produces amino acids that can be used in the synthesis of the proteins required for formation of seedlings that are young plants. Figure 2.1 is an incomplete diagram of the molecular structure of the smallest amino acid, which is glycine. Each molecule of glycine has 2 carbon atoms. Complete figure 2.1 to show the molecular structure of glycine. So you have 2 of the carbon atoms drawn already. The terminal carbon atom is part of an alcohol group. So you'd have carbon double bonded to an oxygen and single bonded to an OH group. And on the other end, you'd have an amine group. So here's the alcohol group, and here's the amine group. 3. Starch is a mixture of 2 different molecules. Name these 2 molecules, which would be amylose and amylopectin. 2 of the enzymes synthesized in the barley seed are alpha amylase and maltase. These are involved in the hydrolysis of stored starch during seed formation. In food industry, the starch extracted from barley seeds, also known as barley starch, is used in the production of sugar syrups. Figure 2.2 summarizes the reactions catalyzed by alpha amylase in the production of maltose syrup and by maltase in the production of glucose syrup. So alpha amylase breaks down starch into maltose, and then maltose is broken down by maltase into glucose. B. Some of the substances shown in figure 2.1 are listed in table 2.1. Complete table 2.1 to identify which of the terms polysaccharide, monosaccharide, and macromolecule apply to each of the substances listed. Use a tick if the term applies and a cross if the term does not apply. Put a tick or cross in every box. So glucose is not a polysaccharide because it is a single sugar unit, and therefore it is a monosaccharide, and which is the same reason it is not a macromolecule, because that would be a large biological molecule. Similarly, maltase is an enzyme, and therefore it is not a polysaccharide, it is not a carbohydrate, it is not a monosaccharide either, because it's not a single sugar unit. However, it is a macromolecule because it is a large biological molecule. Remember, maltase, an enzyme, would be consisting of at least a single polypeptide. Maltose is a disaccharide, so not a polysaccharide, not a monosaccharide either, nor is it macromolecule. Starch is a polysaccharide as it consists of many monosaccharides, but it is not a monosaccharide and it is a macromolecule because it is a large biological molecule. When producing sugar syrups, there are advantages in using enzymes extracted from microorganisms. For example, some enzymes extracted from microorganisms are heat stable, so they would not be easily broken down by heat. Heat stable enzymes are used to increase productivity because the reactions can be carried out at higher temperatures. Increase in productivity is an advantage listed. C suggests one other advantage of using enzymes obtained from microorganisms rather than enzymes extracted from barley seeds in the production of sugar syrups. So, as the question mentions, these enzymes can be used at higher temperatures, which means higher temperatures of operation allow greater rate of reaction. The figure 2.3 is a graph showing how the activity of amylase, alpha amylase, extracted from barley seeds changes as the temperature increases from 10 degrees Celsius to 66 degrees Celsius. So you have temperature range on the x-axis and percentage enzyme activity on the y-axis. One explained the effect of temperature on the activity of amylase, alpha amylase, extracted from barley seeds as shown in figure 2.3. So this time you don't need to write down any descriptions or quote any data from the graph. However, you do need to explain the graph, the shape of the graph. Increasing the temperature increases the kinetic energy of the molecules, and so they move faster and therefore collide faster, which leads to a higher rate of formation of enzyme substrate complex as enzymes collide with their substrate. 50 degrees Celsius is the optimum temperature of the enzyme, giving the maximum rate of reaction, and after 50 degrees Celsius, the percentage activity of enzyme starts to decline, and that's because beyond the optimum temperature, heat starts breaking down the hydrogen bonds that hold the shape of the active site or the tertiary structure, and as a result, the enzyme denatures, allowing no more enzyme substrate complex formation, and therefore the rate of reaction would also decrease. To sketch on figure 2.3 the curve that would be obtained using alpha amylase enzyme that is heat stable. If it's heat stable, that would mean it would denature at a higher temperature. And as you can see, this enzyme denatures right after the optimum temperature. That means if the heat stable enzyme denatures at, let's say, around 80 degrees Celsius, it would also have an optimum temperature that is greater than 50 degrees. And so my graph would look something like this. 3 T helper lymphocytes and Leydig cells are two types of mammalian cells. The main role of T helper lymphocytes and Leydig cells is to synthesize and secrete cell signaling molecules, where T helper lymphocytes synthesize proteins such as cytokines, and Leydig cells synthesize the steroid or lipid, which is the hormone testosterone, from cholesterol. Leydig cells also synthesize cholesterol. A state one way in which cytokines are involved in immune response. The cytokines released by T helper lymphocytes stimulate macrophages to carry out phagocytosis more rapidly. B figure 3.1 shows part of a mammalian cell. One state with reasons whether figure 3.1 shows part of a Leydig cell or part of a T helper lymphocyte. To answer this question, you need to relate the diagram with the function of the cells in the question. T helper lymphocytes synthesize proteins. Protein synthesis requires ribosomes, which are found in the nucleus and also on rough endoplasmic reticulum. And since neither of those is really visible in the diagram, this indicates that the cell is a Leydig cell. To underline the correct name for the type of image shown in figure 3.1 and explain your choice. In this diagram, these structures, I'd rather say, one over here, this one, and also these two are mitochondria. These structures have membranes which are folded to form projections, and those would be cristae found in mitochondria. Also, the internals seem to be similar, but the shape is different, which also indicates that these structures are mitochondria because they can change their shape. And because those cristae or internal structures are visible, this would be a transmission electron micrograph. Two testosterone molecules and cytokine molecules are transported in the circulatory system to reach their target cells. Testosterone molecules are able to enter target cells and bind to receptors within the cytoplasm, so that those receptors could be present in an organelle in the cell, in the cytoplasm of the cell. One outline one way in which testosterone molecules could enter their target cells. Testosterone molecules are hydrophobic in nature, and that means they would be soluble in the hydrophobic interior of the cell surface membrane. Therefore, they do not need any transport protein molecules, so their mode of transport would be simple diffusion without using any proteins from an area of higher concentration to an area of their lower concentration. Cytokine molecules are not able to enter target cells. Suggest and explain why cytokine molecules are not able to cross the cell surface membrane to enter their target cells. Alright, if cytokines are not able to cross the hydrophobic interior of the cell surface membrane, that means they are polar molecules. And polar molecules require transport proteins. If polar substances are not able to cross the membrane, that means a transport protein specific for these substances is not present in the membrane, and that seems to be the case for cytokine molecules. For A, using a light microscope at a magnification of 400 times, it is possible to identify different types of blood cells in prepared slides of mammalian blood. Figure 4.1 is a key to identify different types of blood cells in prepared slides of mammalian blood. In Figure 4.1, letters C, D, E, and F represent four different types of blood cells. One identifies cell types C, D, E, and F in Figure 4.1. C corresponds to the description of 1b, which says nucleus is absent, and nucleus is absent from red blood cells. Now 1a says if the nucleus is not present, you'd go to 2, and the first option, which corresponds to cell D, says a large nucleus, a large rounded spherical nucleus, and that would be a lymphocyte. Now if the nucleus is not rounded, you'd go to 3a, where the nucleus is kidney shaped, and that would be a monocyte. Last but not least, you'd have 3b, where the nucleus of the cell is lobed, and that would be neutrophil. To explain why blood taken from a person with an infectious disease may have a different number of white blood cells compared with blood taken from a healthy person. Infections caused by foreign antigens stimulate immune response, and this might lead to clonal expansion, for example, that increases the number of B lymphocyte cells by mitosis so that they are able to synthesize and secrete a large number of antibodies to fight off that infection. As part of a research project, a team of doctors and scientists carried out tests to investigate the effect of altitude on the ability of blood to carry oxygen. They assessed the changes in the oxygen carrying ability of their own blood as they walked from 1530 meters above sea level to 5700 meters above sea level over a period of 14 days. 5700 meter above sea level is an extremely high altitude. Table 4.1 is a summary of some of their results, and part B asks you to compare results for day 1 with the results for day 14 in table 4.1 and explain the results shown for day 14. Percentage saturation of hemoglobin with oxygen in blood leaving the lungs, as you can see, is much higher at day 1 at a lower altitude than the percentage saturation at day 14 at 5700 meters of altitude. And that is so because at higher altitudes the partial pressure of oxygen is lower, which reduces the affinity of hemoglobin for oxygen, and as the response of body to that change to that change, the number of red blood cells produced increases to compensate for the loss, and this leads to greater number of red blood cells in every 100 centimeters cube of blood. And as you can see, the volume of oxygen delivered to tissues by 100 cm cube of blood does not change much. It's only a difference of 0.1 cm cube. That's because in the same volume of blood at higher altitudes, the number of red blood cells increases. And although the percentage saturation of red blood cells decreases, the increase in number of red blood cells makes up for it, and therefore the oxygen supplied to the tissues remains almost the same. See, the results of the research project have led to an improved understanding of diseases such as sickle cell anemia. Explain why sickle cell anemia is an example of a disease. It is a disease because it is an illness of the body, and that leads to reduced transport of oxygen in the blood due to difference in red blood cells. Each meristem cell in a leaf bud is able to grow and divide by mitosis to produce two daughter cells that are genetically identical to each other and to the original dividing cell. A figure 5.1 lists the stages in the mitotic cell cycle of a meristem cell. So you go through G1, S phase, G2 phase, and then followed by all of the phases of mitosis and lastly cytokinesis. One, outline and explain the events occurring during S phase, metaphase, and anaphase of the mitotic cell cycle that are important in the production of genetically identical daughter cells. Genetically identical cells require identical sets of chromosomes, and so in your answer, you need to state how all of the steps shown in the question ensure that does happen. And so S phase is semi-conservative DNA replication phase, where genetically identical sets of chromosomes are produced. This question is only a 4 mark, so I won't be writing explanations for all of these, since just stating their function would give 4 marks, as I wrote an explanation for the first stage. However, you can write explanation for any one of the three stages. During metaphase, the chromosomes are aligned along the equator. During anaphase, the sister chromatids of each of the chromosomes along on the equator spindle are pulled apart as the length of the spindle fiber shortens from the end of the centriole and the centromere end of the spindle. Two, immediately after cytokinesis, daughter cells are not identical even though they are genetically identical. Suggests a reason why daughter cells are not identical immediately after cytokinesis. So they are genetically identical because they have the same set of DNA. However, the entire cells which contain organelles other than the nucleus are not identical because cytokinesis by definition is division of cytoplasm, and that is not guaranteed to be there is not guaranteed to divide all of the organelles equally. B. Some of the cells resulting from mitotic division in the young leaf from elongated cells that develop into xylem vessel elements. The xylem vessel elements are joined end to end to form xylem vessels. One suggests the structural changes that occur when elongated cells develop into xylem vessel elements, and explain how these changes help xylem vessels to perform their function in transport. So to write down the changes that occur, you need to use the differences between xylem vessel elements and a young leaf cell as reference to answer this part of the question. In xylem vessels, there is no cytoplasm or any other organelles. The end walls are broken down, cell wall is lignified, and this ensures that the lumen of the vessel is hollow to reduce the resistance to water flow, which in turn increases the rate of water flow in the xylem vessel. 2. Figure 5.2 is a plan diagram of a transverse section through a dicotyledonous leaf. Add a label line and the letter X to figure 5.2 to identify the location of the xylem tissue. This part over here is the xylem tissue. This structure represents vascular bundle. 6. In March 2019, a tropical cyclone in the southwest Indian Ocean caused widespread flooding in a number of countries. The flooding and the damage caused by the cyclone meant that many people were at serious risk of cholera. After the natural disease occurred, many different areas reported outbreaks of cholera. Within a short time, the disease had spread widely and large numbers of people were affected. A cholera is caused by a bacterial pathogen. Name the bacterial pathogen that causes cholera. It is caused by Vibrio cholera. B. Suggest and explain why the people affected by cyclone were at serious risk of cholera. Cyclone led to water flooding into areas, and this water was contaminated with the bacteria, and people could get infected by transmission of bacteria from water to these people by, for example, them handling utensils, cooking utensils, without washing their hands after using contaminated water, which is how they could have ingested the bacteria. In addition to standard treatment for cholera, antibiotics are recommended for people who are moderately ill or seriously ill with the disease. One doxycycline One doxycycline is one of the antibiotics used for the treatment of cholera. Doxycycline enters the pathogen and binds to one of the subunits of the bacterial ribosomes. This prevents growth and reproduction of bacterial cells. Suggest and explain how binding of doxycycline to ribosomes stops growth of bacterial cells. If the antibiotic binds to the ribosomes, that means mRNA and tRNA are not able to bind with the ribosome. That could possibly mean the binding sites of mRNA and tRNA are somehow blocked, and so they would not be able to bind with the ribosome, which means that translation of the process of the protein synthesis would not be carried out. And as a result of that, growth would not be possible since growth requires proteins. 2. Penicillin, which used to be prescribed for the treatment of cholera, has a different mechanism of action to doxycycline. State which part of the bacterial cell is affected by the action of penicillin. It affects the cell wall. Mozambique was one of the countries badly affected by the cyclone as part of the effort to prevent a greater number of cases of cholera from occurring. Two different approaches were taken. Approximately 900,000 doses of oral cholera vaccine were sent to Mozambique, and a large scale vaccination program was organized. Medical centers were set up in Mozambique to treat people with cholera. Suggest and explain how the two different approaches help to prevent a greater number of cases of cholera from occurring. So, vaccine would stimulate an immune response involving synthesis of memory cells, which would provide long term immunity. And that means if herd immunity is achieved among the population, a transmission cycle would be broken down, and therefore it would reduce the chances of greater number of cases of cholera. Similarly, medical centers could provide oral rehydration therapy, which would cause recovery of many of the cases. And these centers could also take measures to prevent transmission of pathogen by restricting contact between infected and non-infected people. And so that would also reduce the risk of the number of cholera increasing. This is it. We are done with this paper. Thank you for watching.